going to talk about the germinal centers in health and disease. And probably many of you may not have information about the germinal centers, and the term itself might not be very popular and common. So basically what I want to do is to introduce this field of immunobiology and show you what it does in the context of regulated immunity and in dysregulated immunity in disease conditions. Okay, so the outline of my talk, we will start by asking how famous is the field of germinal centers. Then we are going to go to the definitions and milestones in the history of this field. Third, we're going to talk about the function of the germinal centers. What do they do? What happens in the germinal centers? And we will end with the germinal centers in diseases. So how do they contribute to disease conditions, and how they regulate the prognosis of many diseases. Okay, so how famous is the field of germinal centers? Now, if you search in the PubMed for the word germinal center versus other immunological phenomena and scenarios like immunization, allergy, infections, immunodeficiency, cancer, inflammation, autoimmunity, what you will find is that we're talking about thousands here, 100,000, 1 million if we're talking about infection, beyond 2 million if we're talking about cancers, but we're just getting 8,000 when we search for germinal centers. So this field is to some extent restricted. Do we know about the germinal center cells? How much do we know? If you again investigate the PubMed and look at the germinal center cells, for example, we have follicular dendritic cells versus dendritic cell, tangible body macrophages versus macrophages, follicular helper T cells versus T cells, germinal center B cell versus B cell. So these are the germinal center cell compartment versus the general immunology compartment. Again, a big difference. We have a maximum of like 2,000, 3,000 now follicular dendritic cell publications, whereas when you go to the conventional dendritic cells, we're talking about 100,000 publications. Tangible body macrophages, I would say you will have very few publications on these macrophages which reside in the germinal centers, whereas you will find 250,000 macrophages in the classic context of macrophage biology. Follicular helper T cells, maybe 1,000 publication versus 500,000 for T cells. Germinal center B cells, maybe 2,000 publications versus 250,000 for B cells in general. So the field is to some extent very specific, and having not too much information about it is expected. And again, the community, the germinal center community, and this is the conference that we usually attend, were less than 200 survivors on the face of Earth working in this immunobiology field. We gather in the Germinal Center Conference every 3 years, and we know each other personally. We have our labs that conduct research on the germinal center. Okay, so I introduced the fame of the germinal centers that we know that's not that famous. So what are the germinal centers? The germinal centers exist in secondary lymphoid tissues. These are histological areas in secondary lymphoid tissues. You can see them in the tonsils, you can see them in the lymph nodes, in the spleen, in pyrus patches. You don't find them in the primary lymphoid tissue like the thymus and bone marrow unless there is a disease. So in Graves' disease, for example, in the thymus, you can get some tertiary lymphoid structures with germinal centers. And again, in autoimmune conditions, there is a lot of ectopic lymphoid neogenesis with germinal center formation in rheumatoid arthritis, lupus, Sjogren's syndrome, and that sort of thing. Okay, if you have a cross section, a sagittal section in the lymph nodes, and you look at the different regions in the lymph node, you will find a cortex, deep cortical units, and the medulla. Okay, now the cortex has the B cell follicles. So the B cell follicles exist in the cortices of the lymph nodes. In the deep cortical units, you have the conventional dendritic cells and the T cells. They exist in this area here. And in the medulla is mainly the site for plasma cells and macrophages. And within the B cell follicles, you will find a stromal network of cells. This is the follicular dendritic cell. And associated with these follicular dendritic cells are the germinal center B cells, the green ones here. And they are all surrounded by a resting mantle of resting B cells that haven't been activated, CD19 positive, and they are GL7 negative. They are not activated B cells, called the mantle B cells. Okay, now these three zones together, the mantle of resting B cells, the follicular dendritic cell zone, which is called also the germinal center light zone, where the B cells interact with the antigens, and this green zone here, which is called the germinal center dark zone, where B cells proliferate and experience mutations. That whole thing together is the germinal center. So by definition, germinal centers are histologically identifiable. This is histology, guys, or histopathology. You have to see it with your eyes in order to say whether there is a germinal center or not. Germinal centers are histologically identifiable T cell dependent sites within activated secondary B cell follicles where immunoglobulin affinity maturation and the memory generation occur. Germinal centers are seen 3 days after antigenic stimulation. So if we look at this definition, we find number one, these are histologically identifiable entities. So you have to have an organized tissue, integrated tissue to say, okay, I have a follicle here and I have an active germinal center within this follicle. Number two, it's T cell. Number two, it's T cell dependent. So being T cell dependent means that when we have T cell deficient models like SCIDs or like nude mice, you don't expect that you have germinal centers in these mice. Although we showed later that we can induce some germinal centers in T cell independent fashion. What do they do in this histological domains? You class switch the immunoglobulins from one class to the other, from IgM to IgG, from IgM to IgA, for example. You increase their binding affinity to the antigen, affinity maturation, and very important, you generate the memory B cells. So the cellular memory of the humoral immune system is generated here, long-lived plasma cells and memory B cells. Okay. If we follow the milestones of the description of germinal centers, it started with the description of lymphoid follicles in 1687. Then the first description of the germinal centers was in the 1800s. Then you can go with the eminent British immunologist John Humphrey, who launched the first germinal center conference. Then my mentor in the United States, Professor Anders Sakal, was the first to describe the presence of follicular dendritic cells and their histology. Then the other mentor for my postdoc, Professor John Tu, gave the first systematic nomenclature of dendritic cells. Then the story went from that until 2007, when I and Dr. Tu described the first licensed in vitro germinal center model, or called the modular immune in vitro construct, and this was licensed by Vax Design Corporation. One thing that I want to mention here is that the name for the follicular dendritic cell, which is the main stromal cell in the germinal centers, has been coined in the 80s by John Tu and Ralph Steinman. And I think you know that Ralph Steinman received the Nobel Prize in 2011. And unfortunately, he received the prize after passing away. Although he was nominated for this prize for long, but it took some time because there were a lot of debate whether there are something called dendritic cells or not. The macrophage biologists always believed that dendritic cells are a subset of macrophages, until he finally proved that no, dendritic cells is another kind of cells that is not macrophage in their biology. Okay, so the in vitro germinal center that I developed with John Tu, it includes all the cellular elements of the in vivo germinal centers, where you can predict the immune response to a vaccine, biologic drug, biological warfare, environmental irritants, cosmetics, anything in a test tube. So you are moving from clinical trials into a clinical trial in the test tube, and it really helps to dissect all these mechanisms in vitro, rather than going to the human, where you cannot do in vivo experimentation on the human immune system. Okay, what we do in this, we isolate the different components of the germinal center. So this is, for example, the IgD resting mantle here. CNA42 stains the follicular dendritic cells in the tonsil. So this is a tonsillar section. So we take the follicular dendritic cells, we isolate them. We have a way to isolate these cells. We are one of the very few people who can do this, because it's a tedious, long process. And then we take also the T cells, and we co-culture these cells together at specific ratios, with a specific complementation of the cultures with sera, complement, and other factors. Then in 5 days, you can get a recognizable germinal center reaction in the tube. So you can see that, and we even analyzed these in vitro germinal centers in the culture plates, and they show areas of extensive proliferation and a kind of miniature dynamics between the light zones and the dark zones. This work has been patented in the States and has been used in 40 other patents, including methods for determining the immunogenicity of antigen, artificial lymphoid tissue equivalent, automated artificial immune systems, and disease model incorporation into an artificial immune system. And we can support any research in the field of B-cell biology, humoral immunology with this in vitro germinal center reactions. Okay. Last thing I'm going to talk about is the contribution of the germinal centers to diseases. Now, as we said, germinal centers are formed of B cells and a specific B cell type called the germinal center B cells. The unique feature of the germinal center B cells is that these are non-circulating cells. So you can tell me that I'm going to take a blood sample and look for the germinal center B cells. Germinal center B cells do not exist in the blood. Germinal center B cells either emerge from the germinal center as memory B cells, plasma cells, or they die in the germinal center. Okay? T cells, and we have follicular helper T cells, and these are very critical to provide B cell co-stimulation. We have a specific type of dendritic cells, follicular dendritic cells, and the unique feature of follicular dendritic cell, they are resident. They don't migrate. They don't have motile behavior in the mature form. And finally, the macrophages, and we have a very specific macrophage type called the tangible body macrophages. These cells are responsible for clearing the dead B cells in the germinal centers. So B cells, when they don't survive the germinal center reaction, they die and they are cleared by the macrophages. Okay, functionally speaking, the dendritic cells come with the antigen, they present the antigen. This is what happens in the germinal center reaction. They present the peptides to the T cells, they provide co-stimulation. You have a clonal expansion of the T cells in the deep cortical units. Then some of the T cells interact with the B cells at the follicular margin. Then you have a wave of B cell proliferation, giving you some IgM at the very beginning of the response. This is extrafollicular activation of the B cells, and you get IgM in the circulation. In 3-4 days, you can see IgM in the circulation. Now you take this IgM, and some of it traps the antigen on the surface of the follicular dendritic cells. Then the B cells start to interact with the antigen and test the affinity of their receptors to the antigen on the FDC. If the affinity is good, if the affinity is bad first, then this clone is usually deleted and die and cleared by the tangible body macrophages. If the B cell receptor has a better binding capacity to the antigen on the FDC, this B cell clone is going to survive and is going to be given bafinapril from the FDC. These are cytokines that provide survivability to the B cell. Fine. And they go into this rounds of mutations and testing the generated affinity of the receptor in cycles until in 2 weeks' time, you end up with the outcome of the germinal center. You have class switch recombination from IgM to other subclasses, to other classes like G and A and E. There is somatic hypermutation providing affinity maturation and secretion of high affinity antibodies. So the affinity, the binding capacity of the antibody increases, and you have the generation of the long-lived plasma cells that home to the bone marrow, and you generate the memory B cells. So these are the four outcomes of the germinal center: class switch recombination, affinity maturation, and generation of memory. And as we said, this is a T-cell dependent response, and macrophages contribute by clearing the dead B cells from the reaction in order not to allow self-antigen to be exposed to the B cell receptors that may precipitate autoimmunity. Again, the outcome of the germinal center, it's AID dependent. It has to be induced by T cells, and it generates the different outcomes we mentioned. Okay, this is the AID. This is the enzyme responsible for the two major events in the germinal center B cell: class switch recombination and somatic hypermutation. It's a 24 kilodalton protein that you can easily detect in the germinal center B cell. Why do you need class switching quickly? Because the IgM won't help you too much. IgM has a limited serum half-life, like 3-4 days. You need to switch to IgG in order to provide longer antibody life, to 3 weeks, for example. IgM cannot go to the tissues. IgG can cross the endothelium and goes to the tissues to combat infection. So the diffusion of the IgG is better than the IgM. IgM is very good at complement fixation at the initiation of the immune response, but it won't help on the long term. Why do we need somatic hypermutation? We need somatic hypermutation because we need high affinity antibodies. High affinity antibodies are very important in things like lethal toxins of bacteria. Botox, for example, the lethal dose of Botox is 1 to 2 nanogram per kilogram, okay? Unless you have very high affinity antibodies that can block the catalytic site of these enzymes, the patient may die. You need high affinity antibodies for COVID-19 in order to neutralize the virus and prevent it from binding to the ACE2 and entering the host cells. Why do we need memory? We need memory because you don't want to mount a delayed response each time you are exposed to the infection. You need a more potent, rapid response within a week rather than 2 to 3 weeks when you are secondarily exposed to the infection. So you need this kind of memory. Okay. The memory, which is the last thing that I'm going to talk about, do we develop memory for vaccines, for infections, that sort of thing? There are two major models for memory generation in immunology. One that has been coined by Rafi Ahmed, and this guy worked mostly in mice. And that's mice will have a lifespan of 2 years, something like this. So he postulated that we have a long-term, long-lived plasma cells that can survive for years. When Antonio Lansvacchia tested this in the humans, the story turned out to be different a little bit. Days after boosting, you may stay until 120 days, 240 days, something like this. But to have long-lived plasma cells forever, this is to some extent not the standard in humans. In diseases, germinal centers have been associated with many, many disorders. autoimmunity, chronic inflammatory conditions, infectious diseases, and tumors. So, for example, in autoimmune disorders, in rheumatoid arthritis, you have up to like 65%, not even 35, 65% synovitis associated with the development of ectopic lymphoid structures and germinal centers. Hashimoto, 100% of patients, they do develop lymphoid, ectopic lymphoid structures. Same story will go with cryptogenic fibrosing alveolitis. Same story goes with Helicobacter pylori infection of the stomach. You may go up to 100%. Ductal breast carcinoma, 100% association with the development of germinal centers. Okay, what happens in these germinal centers in autoimmunity? Most of the germinal centers are aggravating factors, pro autoimmunity. They aggravate the autoimmune disease. The association with tumors is to some extent different. Development of ectopic lymphoid structures associated with germinal centers is thought to be a favorable scenario that is associated with good outcome. So the final slide, germinal centers, these are activated B cells inside the B cell follicles in the cortices of the lymph nodes. Most of these spaces are filled with proliferating B cells, very little stromal cells, the follicular dendritic cells, not like the paracortex and the deep cortical units. Inside these germinal centers, these hotspots are the places where you induce class switching, you induce affinity maturation, and you generate memory for the immune system. And thank you.